Good afternoon, everyone. You might have heard a lot about Mars lately, and one of the reasons for that is that NASA announced that they wanted to send humans to Mars in the 2030s, and SpaceX, which is a private rocket company, wants to send people there in the 2020s. It does not mean that it will happen in those exact time frames, because delays are common in space projects, maybe even more than in biotechnology projects. But I'm quite confident that we will have the technologies ready to land on Mars within the coming decades. Now, getting there is only one of the issues of the Mars mission. And among other things we need to solve, there is a survival issue, survival. I mean, we need to keep people alive. We need to provide them with food, oxygen, water, and a few other things. But I will come back to that later. Now I would like to speak about another issue, which is a psychological issue. The thing is that astronauts going to Mars will be isolated. I mean, there is when you're far away from Earth, there is a significant delay in communication. And when you're on Mars, it takes between 3 and 23 minutes roughly to send a message. So it's even worse than trying to have a Skype meeting with the Wi-Fi of a university. And people will be confined. They will be in tight quarters for 2 years and a half, maybe more. And they will not have the ability to go outside, to go in the open air. And the question is whether the psychological difficulties of such a mission will be such that astronauts will not be able to perform at a high level, or that they will not be able to work together as a team. And to answer these questions, NASA is funding a program called High Seas, which is led by the University of Hawaii, where crews of 6 people are living isolated in a dome on a volcano, which is an 11-meter in diameter dome. And they are living there basically as if they were on Mars. I mean, they are isolated. There is a 20-minute delay in communications, which means that every time you want to send an email or someone sends you an email, the message takes 20 minutes to arrive. And they are confined. They never leave the dome without a spacesuit. And the longest of those missions lasted for a year. It was a mission ISS-4. And I was lucky enough, or stupid enough, you might think, to be one of them. And the 6 people spent 1 year there, which means that for a year they didn't see or didn't talk to anyone besides the crewmates, and they were never exposed to fresh air. And we entered the dome on August 28, 2015, and it was quite an intense moment, because when we were outside, there was a lot of noise, a lot of agitation, a lot of people, and then we entered the dome and we closed the door, and suddenly it was quiet. And we looked at each other, and we looked around us, and we thought, well, from now on and for a year, it will be just the 6 of us and always in this place, or in the desertic landscapes around. This, by the way, is a main room. That's where we're exercising, eating, working, and generally spending time together. And the doors you see on the top were our bedrooms, which were not very big, as you can see. But they were quite comfortable, and we tended to like them simply because it was one of the only places where we could isolate ourselves. Well, there was the bathroom, of course, but you would make enemies if you were spending too much time there. And when I say that we were isolated, it's actually not completely true. We were isolated in the sense that people could not see us, when we were in the bedrooms. But there was basically no sound insulation. The walls were not soundproof, so I could always know, just by listening, where my crewmates were and what they were doing. Which is a bit ironical, if you think about it, that I was never so much surrounded by people than when I was in isolation. This is our kitchen, and which is pretty much like a normal kitchen, but the big difference is that all the food we have is shelf-stable. Well, mostly because, as far as we know, there is no supermarket on Mars. Here you have a few examples of the kind of food items we had. You can see dehydrated chicken cubes, freeze-dried fruits, and dried vegetables, and a few other things. So maybe it doesn't look very appealing to you, I don't know. It's quite okay, you get used to it. And we also had a little bit of fresh food, because we brought some microbes with us and we were able to turn flour and dehydrated milk into bread, into yogurt and cheese and a few other things. And we also brought some seeds with us, and we grew some greens with basically what we could find and some sources of nutrients which are a bit experimental. But to be honest, the quantities were not huge. It was the fraction of fresh food we had was quite tiny. This, just to give you an idea, is our first harvest of cherry tomatoes. It was pretty good, but well, there was still room for dessert. This is our bathroom. As you can see, there is a shower, which is quite a luxury when it comes to space exploration. But water will be quite precious on Mars, and so was it in the dome. So we took about, at most, 2 showers of 30 seconds each a week. Well, next to it you have the bathroom, which is a composting, basically it's composting toilets to save water, which means that you went there, did what you had to do, and microbes would turn that into a compost. And every once in a while, we were opening it, taking the compost out, and cleaning some of the inner parts of the bathroom, which was not the most glamorous part of the mission, but if everything went well, the compost was odorless. Anyways, this was just to give you an overview of our living condition there. But now what we are doing was mostly research. And there was first research for which were test subjects. For instance, we're answering a lot of surveys. Every day we're answering questions about what we did that day, about how we felt, about how we are working together, and a lot of other things. Then we're giving biological samples. We're giving some saliva samples, urine samples, and hair samples. Oh, sorry. And hair samples. Which was quite funny. We all had a shaved patch in the back of our heads, which was not for fashion purposes, but the goal was to quantify some hormone and then basically assess how our stress evolved throughout, during the mission. Then we're also wearing some sensors. We had some sensors around our wrists, around our chest, around our necks, around our neck, which were able to know our heart rate, how we slept, our physical activity, and a few other things. And we had those badges, the kind of boxes that you see around our necks, and those things were able to know who you were talking to, how close you were when you were talking to them, how you were talking to them, and how your body was reacting to this interaction. There were some cameras in the dome, but funnily enough, we felt like the badges were watching us. Then we're testing some technologies which were developed for Mars exploration, and in particular technologies which are developed to attenuate the effects of isolation and confinement. And some of them, for instance, were based on virtual reality, with some glasses that gave you the impression that you were in nature or in a city or somewhere else. But the part that we liked most was research that we're doing outside. Because about twice a week, we're putting on spacesuits and going in the lava fields around the dome for various research studies on the lava fields or underneath the lava fields in lava tubes. So if you don't know what lava tubes are, they're basically some kinds of tunnels in the lava fields. And the way they form is basically that you have a flow of hot lava, and on the outside it cools down in contact with the air, and it forms a solid shell. But the inside is still hot and flows, keeps flowing. So in the end you end up with some kind of an empty tube. And they can be very long. Sometimes we're walking underground for hundreds of meters. And it was most of the time pitch black. Like on this photo, it looks. Well, on this photo, it looks like there is some light, but it's actually the flash from the camera. Okay, in reality, we were just exploring with our flashlights, and it was very, very interesting, because you never knew what you were going to find next. You know, sometimes you were in a huge room, and then you arrived at the end of it, or at least that's what you thought, and you found a hole in the ground, and then you crawled through it for a few meters, and then you arrived in a new room, which could be like even bigger than the first one, or that could be very narrow on the contrary, and you had some rocks with crazy shapes, crazy colors, and some geological features that you didn't even know existed. And the reason why we are interested in those lava tubes is because there are some on Mars, which can be much bigger because of the lower gravity, and they offer a natural shelter against radiation that we could use. And it's also an interesting place to look for life. Anyway, those were just a few examples of the studies for which we are research subjects. But then there was also the research that we brought with us. We all had our own research projects. There was a biology lab in the dome, and I was mostly conducting some space-related biology research. And I'm going to tell you a little bit about it. Oh, and by the way, if you've been wondering the past 20 minutes why I'm telling you about Mars exploration in a meeting about biotechnologies, that's where it starts making some sort of sense. But before that, I would like to remind you about the issues I brought earlier related to Mars exploration. And one of them was surviving. Well, the thing is, if we want to go to Mars, it's not just to leave a footprint and plant a flag, or to show that one country has a bigger rocket than another, but it's to conduct scientific research. And if we want to conduct scientific research there, search for life, for instance, we want teams to spend extended periods of time on site. Maybe something like we have in Antarctica, a permanently habitated base. The problem then is that if you want people to be there, you need to provide them with a lot of things. You need to provide them with food, with oxygen, with water, with coffee if there are scientists, and a few other things. And it quickly amounts to let's say, tons and tons of resources just for a year and for a crew of 4 to 6 people. And given that sending a payload to Mars is very, very expensive on a per kilogram basis, it very quickly becomes an economist's nightmare. But there might be a solution. And I'm going to borrow a metaphor from an aerospace engineer, Robert Cyprien, which is that in the 19th century, the British Army wanted to explore the Canadian Arctic. And so they sent huge warships which were full of coal, full of food, full of everything that people need to survive. And for years, they fought against the ice sheets until they ran out of resources and they died, or they came back if they were lucky enough. But at the same time, you had some small groups of explorers who had basically no money. And they looked at what the locals were doing, and they started traveling on the ice using dog sleds. And they were hunting and fishing to feed the dogs and feeding themselves. And the funny thing is, they ended up exploring much more than the huge warships. And that's because they were traveling light and using what they found on site. And that's basically what we need to do on Mars. Now, how can we do that? Well, an interesting way could be to use microbes. I mean, we've been using microbes on Earth for centuries, or even millennia. We've been using them for making food, fermented drinks, drugs, chemicals, and more recently biofuels and biomaterials and so on. And some people here are working on giving those microbes new functions. And I'm speaking about microbes because I'm terribly biased by my background, but I could say similar things about mushrooms or plants. Now the problem is, how do we feed them? Because if we bring from Earth all the substrates we need to feed those organisms, well, the mass problem is moved, but it's not fixed, right? So we need to find a way to feed those microorganisms and plants with what we find on Mars. And Well, the good news is that on Mars you have basically all the elements you need to sustain life. You have carbon dioxide in the atmosphere, you have nitrogen as well in the atmosphere, you have water, plenty of water, and you have basically all the mineral nutrients you need in the ground. Now the bad news is most of those elements are in a form that most organisms cannot use. I mean, heterotrophic microbes can do much with the carbon dioxide in the atmosphere. The nitrogen in the atmosphere is mostly as the nitrogen that most organisms cannot fix. And the nutrients in the ground are poorly accessible to most organisms. So we need something to process the resources we find on Mars and turn them into something that those organisms can use. And that's a solution we're working on in the laboratories of Lynn Rothschild at NASA and Daniel Abili at the University of Rome Tor Vergata. And the idea is to use cyanobacteria. Because some specific cyanobacteria could be fed almost directly with what you find on Mars. I mean, they can fix the carbon dioxide in the atmosphere because they do photosynthesis. Some can fix the denitrogen. And they're able to extract nutrients from rocks on Earth, which are very similar to the Martian ground. And once you have grown cyanobacteria, the idea is to use them to feed other microorganisms and plants, and like this create a small-scale ecosystem, which is based indirectly on what you find on Mars, and that can produce basically everything you need to survive. And well, we've shown, among others, that you can actually use cyanobacteria to produce nutrients for other microbes very efficiently, as in just pure water or nutrients produced by cyanobacteria. And this will be sent to space this year on board a satellite, where you will have basically a microfluidic device with some spores of Bacillus subtilis that will be rehydrated in flight with nutrients produced by cyanobacteria. And, oh yeah, and by the way, the spores of Bacillus subtilis will be then transformed in flight, and the expression of the exogenous proteins will be followed. And the reason for doing that is to demonstrate the feasibility of synthetic biology in different gravity levels, in space microgravity, moon gravity, and Mars gravity. Well, why synthetic biology? Simply because it could greatly improve the process. First of all, you could use synthetic biology to increase the resistance of microorganisms to conditions found on Mars. Probably not to the extent where they could grow directly on the surface, because there is too much radiation of different kinds. The temperatures are too low, and the atmospheric pressure also is too low. But if you can increase the resistance a little bit, you can decrease the level of protection which is needed, and you can reduce the costs. Another thing you can do is improve the ability of cyanobacteria to use the substrates available on Mars, and to use the ability of other microbes or plants to use the nutrients produced by cyanobacteria. It's basically the same thing as you do on Earth when you engineer your producing organisms to make them rely on cheaper substrates. And then once you have a production system up and running, you can use synthetic biology to improve the yields, or you can use it to confer microorganisms new functions. And one of the, well, good opportunities for synthetic biology on Mars, synthetic biologists on Mars, is that some compounds which are produced more efficiently using other methods on Earth could be done much better by microorganisms there. I will give you an example. Imagine that you have a compound which is normally produced by a tree, and you're interested in this compound, but you want to produce it with a microorganism. So you will take some metabolic pathways from the tree, put them in your microbe, and do some tinkering, and then in the end you will have microbe able to produce this compound. You've put a lot of work into it, you're very proud of it, but the problem is you cannot go further if you cannot show that this compound is produced more efficiently using your microbes than it is taken directly from the tree. Well, on Mars there is no tree, so it's already very good if you can show that your microbe can produce compound efficiently. Anyway, NASA is putting a lot of efforts into synthetic biology. NASA, and especially NASA Ames Research Center in California. So to sum it up, the idea is that tons of resources of food, oxygen, and so on could be replaced with very small samples of microorganisms and seeds. And that once landed on Mars, travelers could feed cyanobacteria using what they found on site and use them for making production processes which are almost independent of Earth. Anyway, that's the solution that we propose for the surviving issue. Okay, I'm going to use this one. Oh, okay. The solution was for the surviving issue. I would like to come back to the psychological issue, the staying sane one. Well, I cannot give you all the results of the human factor study from HICES before they are published. But I can tell you that we were able to maintain our level of performances throughout the mission, even in the toughest moments. And I can give you my personal impressions, which is that, well, the psychological challenges of mission to Mars are not real obstacles, and they can be overcome by selection and training. Well, a good sign of that is that, well, 6 people came in, 6 people came out. No less, no more. And yeah, I like this photo a lot because, well, you can see that we are smiling, and the reason for that is that we're in the open air outside and seeing other people for the first time in a year. Anyway, that's the end of my talk. I would like to thank the great people I had the chance to work with in the laboratories of Lynn Rothschild, of Danny Labili, and of the HICES formation. Thank you for your attention. We should have plenty of time for questions. So.
Okay, thank you very much, Cyprien. There's quite a few questions about that. Just on the psychology side of it, a few questions. Were you assessed in any form psychologically before you went in? What makes you an ideal candidate, apart from being able to do the science, for going into something like this? And that's the first question, I suppose.
So the question is, what are the psychological criteria for being a crew member in such a mission? Well, I think one of them is to be, easy to live with, simply because you are in tight quarters with people, and basically they are always there. You can always hear them. If you have an argument with them, you have no way to hide. You must work with them, live with them. You cannot even, open the door and go outside. So I think being easy to live with is very important. Then you have to be relatively optimistic. You must be realistic, but you should not be neurotic. As in, if you will get terribly sad or terribly stressed over little things, then you will, basically not end the year, and you will stress your crewmates.
Did you find any changes in your own psychology when you were in there?
Well, let's say that before the mission, I didn't know how I was going to react. Like in my normal life, like if I spend one day indoors, like for working on an article or something, like in the end of the day, I feel like I have to go outside. I have to go running. I have to go take some fresh air. And I was wondering whether it was going to increase throughout the year. If it was going to, make me feel very uncomfortable as time would go by. But I think the biggest change I saw was the adaptation to this thing. The adaptation, and then the fact that after a while you basically accept the idea that you're always in the same place and that you're always indoors, and you just adapt.
You didn't find anything that you had to readjust to anything after coming out? Like people in prison sometimes have trouble coming away from prison because they have to readjust to being outside.
Well, yeah, readjusting took some time. I mean, no, it didn't really take time to basically get adapted to being outside again, but it took time to getting used to it again.
Just in terms of the science then, this is the last question from me. I mean, most of us are used to kind of working in labs and having access, like I said, to the outside world as we work. So you can say, oh, well, I don't have X, Y, Z. I can just walk out the lab down to somebody else's lab, or I can ring up some person for help. I mean, when you're carrying out this kind of science, and this will be true of anybody who goes to Mars, they're sort of in a position where they have to kind of think of everything before they go, because if they forgot their right pipette, they can't exactly turn around. Did you find that working in there, that sort of became a problem at any stage?
Sorry, whether like the fact that I was doing science in the confined environment was a problem?
Yeah, in terms of that you were that your experiment to some extent, the experiments that you did within there, were they sort of confined experiments? Did you find that you wish you had access to other equipment, for example?
Yeah, the thing is that normally, like in real life, when imagine you have a project, you want to get funding, you write a grant proposal, and you say, I will use this and that and that and that, and it will cost me this amount of money and so on. And well, then reality happens, and then in the end, if you compare the list of what you actually use to what you plan to use in the beginning, usually it's quite different, which is not really a problem. Sometimes you I don't know, you realize that there is something you didn't think about. There is someone spilled your reagent, or you had an idea that came along the way. It's okay in your day-to-day life, but when you're isolated, you have to think deeply before about everything you will need during your year. And well, if you forget something, you have to go without it. And it can be quite frustrating. And another thing is sometimes basically the equipment we had was very limited because we're limited in space, and we're limited in power consumption, because basically the power was coming from the solar panels. And we're limited by mass, simply because during a Mars mission, the payload you could bring is very limited. So sometimes I ended up, centrifuging tens of liters of a solution using a centrifuge that could take only, let's say, 50 mL at a time. And so it took sometimes I just got up at 5 in the morning and did centrifuges all day until 1 in the morning. So yeah, these kind of things can be a bit frustrating.
Yeah. So, Mark.
Hi, Mark Tagney, University College Cork. Fascinating stuff. I have a nerdy biology question or two. So am I correct in thinking that for the 1 year you were inside, none of you got sick? No infection?
Well, the good thing is that since we're isolated, we didn't get the flu, we didn't, if no microbe came in, the common microbes that people get in winter, for instance, there was no way they would get inside the dome. So
as a result, I was wondering, from the samples that were being taken from you and analysed, do you know two things? One, was your immune system different going in and out because it wasn't challenged for a full year? And secondly, your gut microbiome must have been different after than before?
So we don't have yet the results of the microbiome studies. So we did take my samples throughout the year, but they have not been sequenced yet. So we don't have the answer to that yet. And it's actually, I'm very curious to know whether, for instance, the microbiome got homogenized between people or not. So, well, we'll see soon. Regarding the being sick, so we had, I think one person got sick during the whole year, but it was because he got a vaccine shot. But apart from that, nobody got sick.
So based on all this, and you talked about the dates when planning to get to the Mars, do you think that those expectations are realistic at present? Those expectations for when we're actually going to go to Mars. So you hear numbers. I think you raised them years raised at the start. Whether it's from SpaceX or from other funds. Are they realistic, or are they very, very highly planned, or are they more aspirational?
No, I think they're very realistic, technologically. Now the big question is whether we'll have the political and financial support to do it. If we had someone like Kennedy who was saying okay, we're gonna go to Mars in 10 years, and actually putting the efforts into it, it could happen. Now, there is no, I think we lack a vision. Or at least we did lack a vision until maybe 1 year ago. We need the effort to be directed, and we need the funding. So it's doable. It will depend a lot on the support we get.
Okay. This is obviously one project, but you drew up a sort of you had a slide there earlier which sort of showed that sort of almost a plant that you would build on Mars in terms of different parts. Has that sort of been created on Earth in all its forms already?
So are you referring to this?
Yes.
Well, this is an artistic view, so don't imagine it exactly like that. So when we're working on it, we don't imagine something that big.
are all the components for it buildable at this stage? You know what I mean, in terms of, do we have those kind of microbes that will sort of act under those kind of conditions and various things? Or is that, are we still at sort of what stage of development are we at each of those points?
So basically, we know that, well, in theory it works. I mean, we still need to test it using like resources that actually come from Mars, because there are something in the ground, for instance, we still don't know. But from what we know on, of Mars, on what we simulate on Earth or in the space station, basically the concept works. Now it's mostly a matter of making it energy efficient and mass efficient, basically optimizing it so that we can actually turn it into something which is very concrete and, well, realistic with the funding we'll have.
Okay, thank you. Have we any other questions? Oh, we have one up here.
Assume that there is a go to Mars. Would you volunteer to be the first one to be on Mars?
Yes. I mean, I would.
It's a very interesting question because I heard it debated last year when I think people have actually signed up and to what could be regarded as a one-way mission. So it's a good question because it is a one-way mission, possibly at this stage.
Well, NASA is not planning a one-way mission. NASA wants, well, actually, there is no solid plan for a one-way mission. It would be, like, most likely the first mission to Mars will be a mission of like 2.5 years, something like this. So there is, there is this private company, which is Mars One, which basically sells tickets to Mars for basically a one-way trip. And they are planning to fund the whole mission using a reality TV, something like that. NASA has nothing to do with it. And to answer your question, I would go to Mars if I can do something significant, if I can do some scientific research, if I can look for life, if I can... I would even go for a one-way trip if I could go there and prepare the terrain for other missions, which would have a way back. But I would not do a one-way trip to Mars just to do reality TV or something like that.
I suppose the risks are probably always going to be there before you go anyway. In terms of, just go back to your very happy picture there of the 6 of you smiling, are you all best friends now? Or have you vowed never to speak to anybody in that thing again? Because it seems a very intense environment to get to know somebody in a year.
Yeah, I mean, you get to know people very well. Like, there are very few situations where you actually, like, 24 hours a day with those people and you have to work and live and relax and do everything with them. So yeah, it creates some very tight bonds, and I'm still in close contact with most of them.
And where does your work actually take you from here then? So since you've come out of here, come out of this environment, where is your next scientific work going to be based?
Well,
what are your goals, I suppose?
So at the moment, I'm mostly working on, well, I'm working on two things. So I'm working on this, basically, on mostly optimizing the way you can transfer nutrients from cyanobacteria grown in Mars-like conditions to plants and microorganisms, basically the basis for this system. And another aspect of my research is basically to figure out how you can recognize life on Mars. But there are some... Basically, the last mission that directly searched for life on Mars was in the 1970s. You know, and well, the rovers, the Viking rovers, had some tests to try to find some life, and actually one of the tests came positive. The thing is, at that time we didn't really have the basically we didn't have enough knowledge of the Martian ground to completely eliminate for other reasons that could trigger the signal. So basically, the people who got this positive answer about their test for life on Mars are still convinced that they found life on Mars. But many people are saying, no, you know, there are other like products in the Martian ground that could produce a similar answer. And since then, we've sent other rovers on Mars, but they were mostly looking at whether life was habitable. But they didn't look at whether there was life. They didn't look for life directly. But this will end in 2020 when there will be 2 rovers, one from NASA and one from the European Space Agency. and those will look directly for traces of life, for biomolecules. The thing is that we have technologies that, well, we know on Earth can detect life. Like, for instance, you will, send a ladder underground and then look at the signal that is emitted, and you will see, oh, okay, this signal is typical of such molecule which is due to life. But when you're on Mars, you have radiation and you have a different atmosphere and so on, and this can modify the molecules that you try to recognize. So what we're working on now is to figure out how to detect those molecules after they have been exposed to harsh conditions found in space and on Mars.
All right. A completely different question, but I think you're one of the few people that can actually answer that. When you get to ask, like, if you could go to a Soul Island, what will you bring? And I was wondering, what did you actually bring to Mars? And maybe more important, what did you actually forget?
That's a very good question. So, basically, I assume your questions like assume that, okay, you don't include everything that you absolutely need. For instance, my contact lenses, otherwise I will not see anything. That would be the easy answer, but I guess it doesn't make it very interesting. Some things that I didn't have to bring, but that I brought was a ukulele. And which, if you don't know what a ukulele is, it's basically like a tiny guitar with 4 strings. And the reason for this is that, well, it's very small, it's very light, so it's perfect for space exploration when every kilogram counts, And also, it's not very loud. If you want to play the organ, I'm not sure your crewmates will like you very much, and you might end up in the airlock. And well, another reason is that when you take a ukulele out, contrary to when you take a guitar out, people don't have expectations, which is perfect when you have my love and music. So yeah, I think, and more seriously, I think it was very useful to have it there because, sometimes conditions can be a bit stressful. Sometimes, you want to be somewhere else because you've been always in the same place all the time. Imagine you were living in this room or half of this room for a year. After a while, you get tired of looking at the walls. And playing music was a very good way of basically getting rid of the stress and it almost made you feel like you were somewhere else. It basically took you south, and it helped us a lot psychologically. I say us not because they were listening to my music and enjoying it, but because other people were also playing. And yeah. Well, weekends were basically 2 days like any other day. We didn't really have weekly days off. I think we took like 1 day off in the year, and it was not really off because we still had to answer surveys. And I think we were pretty bad at that. Like, we should have forced ourselves to say, okay, I don't know, once a day we don't work at all. Or we just do basically the things that we absolutely need to do, like maintain the habitat and answer surveys. But, well, if you put 6 people who tend to be workaholics at the same place and have no one to, to rationalize them, we don't need to forget about resting, which was a very bad thing when you think about it, because your productivity tends to drop when you do that. But yeah, we should have taken more rest.
Just on that, I mean, what's your contact like with the outside world? In other words, were you restricted in any way from who you could speak to or talk to? Were you able to get emails from all your friends or your family? Or were you restricted in any way?
So, but basically, we could have no direct communication. We could have, as I said, we could have only, we could send emails with a 20-minute delay. So there was no phone calls, of course, no Skype, no WhatsApp or whatever you're using. Like nothing that could allow us to have a direct communication. And we didn't have access to the internet. So we had a few websites we could access, like some static websites, which was basically similar to if we were on Mars, someone made a copy of this website and sent it to a local server. So we could not surf. We could access the NASA website and a couple of other things, and but we could email with our families with a 20-minute delay.
Okay. You just mentioned earlier about political will, or I suppose the financial might that we were required to actually get us to Mars. I mean, is that there? Will it be there? Do you sense that there's a real willpower to get us there?
Well, I think that if we let's say that if well, I don't know, someone like, for instance, the President of the United States, decided to, let's say, okay, we want to go to Mars now, I think that we need to first make it a clear goal, because now some space agencies have many different projects, and I think you need to focus to say, okay, this is a project, this is what we're working on, everyone working in this direction. And then, yeah, maybe increasing the budget would be a good thing. I mean, basically, a mission to Mars has been, well, it's hard to give an estimate because it depends on so many things, but it would be roughly between a few tens of billions to a few hundreds of billions.
Just on the cyanobacteria, I mean, I don't know much about cyanobacteria except for one of the foundations of life apparently on Earth. But is this sort of a very complicated cocktail that people envisage of cyanobacteria? Are they developed in a certain way? Are we looking at genetically altered strains here? What are we trying to achieve with them at the moment?
So, well, the strains we're using now are not, well, we have one strain which was modified. So it's called Power Cell. It's basically a strain of Anabena which is modified to secret sucrose, which could then which can then be used as a substrate by Bacillus subtilis, for instance. But you can also do it with a wild-type cyanobacteria. And then basically it's mostly a matter of optimizing the cultural conditions and optimizing the process by which you will take nutrients and transfer them. So to answer your question simply, synthetic biology and genetic modification in general can improve the process a lot, but it's not absolutely needed to have something running.
All right, I think that's all from me, unless anyone else has any other questions. Oh, we have someone here at the back, sorry.
I was just wondering, because you were there for a year with the other 5 people, what was the makeup of the crew, so the background of the other people who were in the habitat with you? Because you were doing the microbe research, were they doing similar work, or were they?
So there was okay, I will introduce you to the crew members. So basically, this guy here is an aerospace engineer, and he was an interplanetary space flight controller, which means basically that he was remotely piloting spacecraft, like Mars probes and this kind of things. She was a physicist, well, she still is. She's a physicist with a PhD in engineering. And yeah, so he was working with a drone, basically, and he was assessing the feasibility of using a drone for basically inspecting the habitat without going outside, because it's a process to go outside, and it costs oxygen, and it costs lots of time. So if you can use a drone to basically do the daily inspection of the habitat, it could be quite useful, and also to basically improve the safety of the EVAs of the when we go outside. If you can have a drone that monitors the terrain and keep an eye on people, for people who stay inside the dome, it would increase safety. So she was working, the next person was working on how you can extract water from the Martian ground. Yeah, because I said you have water on Mars, but it's mostly water in the form of ice underground or on the ground at the poles. And you have some, you also have some hydrated minerals, but it's quite hard to get them out efficiently. So she was working on a process that basically could take this water out and store it. And the person next to her is a soil scientist, and she's basically been working in extreme environments for years. She's been leading field expeditions in Alaska, for instance. Then there is an architect who's is working in extreme environments, and he's doing his doctorate work on habitats for Mars exploration. So basically he was in the dome to see what could be improved, what should be taken out, these kind of things. Then it's a French astrobiologist, and the person next to him is, she's a medical doctor and a journalist, and she was in the dome mostly for basically looking at some emergency procedures, like basically what you do on Mars if such things happens, because she has some experience in doing that for emergencies on Earth, and she was developing similar things for Mars. She also had a project where basically she used a 3D printer that we had in the dome to print some medical tools. like for making surgeries, for instance. And this, together with protocols that would allow someone which is not, who doesn't have a lot of training in medicine, to basically perform surgery with those 3D printed tools. Fortunately, we did not need that. There is a button on drinks.
I think just before we wrap up, out of curiosity, how many years do you think we are away? I know with Musk talking about getting to Mars by mid-2020s, 2030s. How long would you say your projected time frame is to implement Cyprien on Mars, if all things went to plan and we had Bowie Base 1 on Mars?
Okay. Well, so we hope to have it ready by the time people get to Mars, but it would probably not be implemented right away. It's not like you go there and then directly start relying on it. I think for the first missions, you will bring everything you need. And maybe you will recycle as much as possible your water and a few other things, but you will not depend on technologies which have not been proven in the field yet. So it's feasible, you know, for a 3-year mission, you can bring all the food and everything you need, but then you will. I mean, the way I see it at least is people will go there, and while the consumer resources are bought from Earth, they will start implementing and testing other technologies which rely on local resources. You know, and then perform some tests, maybe get some resources out of it, but not depend on it for survival. And then, well, if things work well, in the next missions, people will rely on it more and more. And then after some time, it will be difficult to give an accurate estimate, but maybe after a few missions, people will slowly start to depend almost exclusively on technologies that are based on local resources.